This is a tutorial video in GCSE chemistry looking at topic 4. This video will focus on acids and bases. In this video we will look at what is meant by an acid and a base. We will look over the pH scale, we will look at strong and weak acids, and finally we will touch on neutralisation. The topic of neutralisation will be covered more in a titration video. In this first part of the video, we're going to have a look at acids and alkalis in terms of their ions and their pH. We're going to have a look at how changes in hydrogen ion concentration affects the pH of a substance, and we'll have a look at dilution and concentration. So what are acids and bases? Well, here we have some common acids and some common bases. We have hydrochloric acid, nitric acid, and sulfuric acid. We also have ethanoic acid or vinegar. On our bases we have sodium hydroxide and potassium hydroxide. In fact, all of the ones listed here are hydroxides. In order to look at these further, we need to have a think about their ions. An ion, as we've looked at before, is a charged atom. This can be both negative or positive. An acid will also produce excess hydrogen ions in water, meaning that the hydrogen ion H+ is found in acidic solutions. Alkalis, on the other hand, produce hydroxide ions. This is a polyatomic ion as it contains two atoms, the oxygen and the hydrogen. Hydroxide ions are negatively charged, meaning that alkalis are negative. A neutral substance, for example pure water, will produce an equal amount of hydrogen H+ ions and hydroxide OH- ions, neutralising each other, causing a neutral solution. In the lab, we use three main acids. All of them contain this hydrogen H+ ion. These are hydrochloric acid, HCl, nitric acid, HNO3, and sulfuric acid, H2SO4. For those of you taking triple science, you will also come across carboxylic acids in the triple part of the organic chemistry unit. The most common of the organic acids, the carboxylic acids, is ethanoic acid, which is better known as vinegar. Bases, on the other hand, contain oxides, hydroxides, and carbonates. They are the opposite of an acid, as we have just looked at. They produce OH- ions, but importantly, do not dissociate into H+ ions. Some bases are able to dissolve in water. These are known as alkalis. Therefore, all alkalis are bases, but not all bases are alkali. An alkali is a soluble base. So what is the pH scale? Well, the pH scale is a measure of how acidic or alkaline a solution is. The lower the pH, the more acidic the solution is. The higher the pH, the more alkaline this is. This also affects the ion concentrations. The more acidic something is, the more H+ ions are present. And the more alkaline a solution is, the more OH- ions are present, but more importantly, the less H+ ions are present. Some examples of different pHs are as follows. Down at pH 1 to 2, we've got things like carbonic acids and stomach acid. Moving up further, pH 3 to 4, we're starting to look at vinegar. pH 5 is rainwater. Neutral, pH 7 is pure water, and then going through alkalis, we've got washing up liquid, we've got bleach, and then things like drain cleaner or very, very alkaline solutions. We can measure the pH of a solution in a variety of methods. One is to use a pH probe. This will give an electronic reading as to the pH. We can also do it by using an indicator. An indicator is a substance that changes colour when it's added to an acidic or an alkaline solution. The ones you need to know are universal indicator, which gives this nice scale from red up to purple, as well as litmus, methyl orange, and phenolphthalein. We will look at indicators in more detail in the titration tutorial. We can also link the dilution or concentration of an acid to the number of hydrogen ions that are present. So the concentration of hydrogen ions in an acid is directly linked to its pH. As we go down the pH scale from 7, there are 10 times more hydrogen ions present each time. So a pH of 6 would have 10 times more hydrogen ions. A pH of 4 would have times 10, times 10, times 10, so 1,000 times more acidic. So as we go down, there are 10 times more hydrogen ions each time. Hydrochloric acid with a pH of 1 is 1,000 times more acidic than vinegar, which has a pH of 4. This means that it has a concentration of H+ ions that is 1,000 times greater. As we go up the pH scale, we get 10 times less hydrogen ions present. So instead of timesing by 10, we can divide by 10 in order to work out the concentration of H+ ions. This increased number of ions in a volume increases the concentration. So the higher the concentration of hydrogen ions, the higher the concentration of the acid is, and therefore the lower the pH. Low pH, lots of hydrogen ions, lots of OH- gives us our high pH. As we looked at in another video where we looked at concentration, a concentrated solution contains a lot of dissolved solute per volume. A dilute solution contains only a small amount of solute, so we can also work out the amount of something that is dissolved into the solution. Concentration is the amount dissolved divided by the volume of solution and is in the units of grams per dm cubed. For example, if 4 grams of a substance was dissolved in 50 centimeters cubed of water, then the concentration would be 80 grams per dm cubed, converting the 50 centimeters cubed to 0.05 dm cubed. As mentioned previously, this concentration calculation is reviewed in a previous video. It's very important to make sure that you always use the correct indicator. Universal indicator gives us a full range through reds to greens and then on to blues and purples. This is very good for finding out the pH of a solution. However, for titrations, which we will look at in another video, it is not as good because it is not a distinct change. The other 3 indicators you need to know are below. Litmus is red in acid and blue in alkaline. Phenolphthalein is colourless in acid but goes pink in alkaline. And finally, methyl orange is purple in acid and green in alkaline. The indicator that we could use to look at neutralisation changes depending on the strength of the acids and alkalis used. A strong acid and a strong alkali can use any indicator, whereas, for example, a strong alkali and a weak acid, we'd need to use Litmus solution. Which begs the question, what do we mean by a strong acid? So, as well as being concentrated or diluted, acids can also be strong or weak. The pH of an acid depends not only on the concentration, but also on the relative strength of the acid. Strong acids will dissociate, meaning they break apart completely when dissolved in water. For example, hydrochloric acid will fully break apart into hydrogen and chloride ions. If an acid dissociates a lot or entirely, then they produce high concentrations of hydrogen ions and therefore have low pH values. Weak acids, for example ethanoic acid, does not completely dissociate into water and therefore has a lower concentration of H+ ions. We can see this in the diagram where we are adding our acid, which has got the proton, the H+ ion and the negative ion attached. As we add it as a strong acid into water, we can see that the 2 ions break apart. For our weak acid, there is some dissociation, However, some of the molecules do not break up, and therefore this would be a weak acid. How you could be asked this in the exam is to compare different acids to suggest if they are strong. Here we have a selection of acids. The lower the pH, the stronger the acid is. Therefore, nitric acid, sulfuric acid and hydrochloric acid would be our strong acids. Boric acid would be our weakest acid here. Another potential question could look like this. So about 1 in 60,000 butanoic acid molecules release hydrogen ions when they dissolve, when nearly all hydrogen iodide molecules do. The first part of this question is to distinguish which acid is a weak acid. Of these two, the weak acid would be butanoic acid, as it has far less dissociation. Which acid will have a higher pH if both are the same concentration? Again, this would be the butanoic acid, as at the same concentration, weak acids have higher pH than strong acids. In another tutorial, we will look at titrations. In a titration, we add an acid to an alkali in order to accurately work out a neutralisation point. In doing so, we can put together a pH curve. On a pH curve, we can record a titration to show us the equivalence point. This is where the pH is changing. In our example, we are adding an alkali to an acid. We start off at a very, very low pH, and as we can see, this gradually increases until we reach a volume added of 25 centimeters cubed. At 25 centimeters cubed, the acid gets neutralized and the pH drastically increases. Past this point, we now have an alkaline solution. So what is neutralisation? Neutralisation is the reaction of adding an acid to a base, or alkali, to form salt and water. For example, hydrochloric acid and sodium hydroxide make sodium chloride and water. In the next two videos, we will look over titrations as well as the reactions of acids to form salts.